right, it's a great pleasure to be speaking to you today. Thank you so much for this very kind invitation. Title of my talk is my seminar is Click Doc Optimize MolModa, a web-driven pipeline for ligand discovery. All right, so just by way of background, my name is Jacob Durant. I'm an associate professor in the Department of Biological Sciences at the University of Pittsburgh. This is what ChatGPT thinks that my office looks like. And in fact, the back wall of my office is not covered in high-tech computer monitors, right? And I don't drink coffee. I prefer my caffeine in concentrated pill form, right? It's much more efficient. But my goodness, it got the overall disheveled look exactly right? It's amazing how well these soulless machines know us, right? So I've had the privilege of interacting with a number of people from the Malaysian drug discovery community, which is why this invitation meant so much to me. I was actually lab mates with Dr. Siti Juso when she was a postdoc in Dr. Romy Amaro's lab at the University of California, San Diego. And then I also had the privilege of collaborating with Dr. Habiba Wahab in 2015. We published a paper together when I was, again, also a postdoc in Dr. Amaro's lab entitled A Virtual Screening Approach for Identifying Plants with Anti-H5N1 Neuraminidase Activity. And so it's been a real fun experience to be invited here and to reconnect with these old collaborators, right? Really delighted to be able to present my work here. All right, so here is a general outline of the workshop that I'm going to try to give. First, I'm going to give an introduction. I just want to kind of give you a sense of my lab's interests, of what motivates our web-centric approach to tool development. Then I want to talk about the MolModa, which is one of our web-accessible tools for molecular docking. And here I'm going to focus mostly on upcoming features because Dr. Juso is going to give a more in-depth MolModa demo afterwards, right? So I'm going to cover, I'll demo a few features and things, but I'm going to focus more on what's coming up in the future. And then also I want to conclude the talk by talking about some other projects we have in mind, particularly how we're going to incorporate lead optimization features into this MolModa framework. All right, so first, by way of introduction, my lab uses computers to study ligand-drug target interactions. And these things can mean slightly different things to different people, right? So I thought we'd just define our terms explicitly so that we're all on the same page. When I say a drug target, I'm talking about a protein with activity that can be altered to improve human health. So you could imagine that DNA or RNA could be a drug target, a lipid bilayer could be a drug target. I'm talking specifically about proteins, right? And then when I say a ligand, I mean a small molecule that binds to a drug target, usually via some well-defined binding pocket on the surface of the protein. And so you can imagine an antibody is a kind of ligand, right? But that's not the kind I'm talking about. I'm talking about these drug-like small molecule ligands. All right, so in terms of what we study, ligand drug target interactions, we study the motions of the binding pocket where that small molecule interacts with the protein. A lot of times the motions of the pocket can impact how well a given molecule will bind there. Then we also study molecular docking. This is a method for predicting how, as you well know, a method for predicting how a small molecule might bind in the pocket. What is its geometry? And then what is its binding affinity? Then we also focus a lot on lead optimization. How can we modify a small molecule to improve its binding by adding molecular fragments, for example? And then we talk a lot about usability. How can we make tools for docking and lead optimization usable so that they can be, have a maximum impact possible by reaching as broad an audience as possible? And so this talk, I am going to touch on molecular docking and lead optimization, but I'm going to talk about them in the context of creating usable tools, right? And we were not going to talk very much about binding pocket dynamics, even though simulating those dynamics is a major part of my lab's work. All right, so what motivates my lab's interest in creating tools that are easy to use and are broadly accessible? Well, it turns out, as many in this room know, many tools for computer-aided drug discovery are very, very difficult to use. And so here, this is an example of Autodoc Vina, which is a very popular program for predicting how a small molecule might bind to a protein. And so to use Vina, you have to first type in, you do it from a command line. You first type in the path to the Vina executable. then you type in the name of a parameter and the value of a parameter and the name of the next parameter and the value of the next parameter. And so it's very difficult to use. And this is a very widely used and important software in the field. And so you might ask yourself, can you believe that something this important is this difficult to use? Well, I could definitely believe it because I have written software that's just as hard to use. So it's not like it's just other people that are guilty of this, right? It's a broad problem that many in the field face. How can we make our tools that are otherwise very powerful, very useful, how can we make them so that they are broadly accessible to a larger audience? And so these kinds of command line tools where you have to type them in, right, they're very powerful. And so I don't want to only talk bad about them. They have some notable advantages. Most of them can run on your local computer. And so you don't have to, if you're running them on your local computer, you don't have to wait for remote resources like a remote supercomputer to become available. So you can run your calculation. If you have a whole bunch of people using the same computer, sometimes that queue time can be very long. And because it's running on your local computer, it protects your intellectual property. And it turns out people in drug companies, for example, care a lot about their intellectual property. They don't want to send their proprietary molecules off to some third-party server and just hope that that third-party server is careful with their data and that there's no data breach or anything like that, right? And then also another advantage of this command line approach is that some computationalists really don't care about graphical user interfaces. They maybe prefer the command line, right? There are weirdos out there who prefer the command line. But as I mentioned, there's these notable disadvantages. They can be very user unfriendly. They require you to download and install separate software. And sometimes the installation is easy, right? But sometimes it requires you to have all the right libraries on your computer. And it can be a pretty difficult. You might have to compile the software to install it, for example. And then some programs, some of these command line tools really only work on certain operating systems. Maybe they only work on Linux, for example, but not Windows. And so there's a lot of disadvantages here. So to address those concerns, a lot of groups have created what I like to call a server app. So here I have a chart of more or less what a server app is. The user visits, goes to their web browser, and then in their web browser, they provide their data. Maybe they upload a structure of a protein, for example, into their browser's memory. And then they also, there in the web browser, specify what parameters they're going to use to run their calculation. And then the parameters and the data get sent to some remote computer resource in the cloud where the actual calculations happen, right? And then once the calculations are done, they're sent back to your browser, and you can then view and save the results there in your web browser. And so the advantage here is that it's accessed online, and so it's usually pretty user-friendly. It's no more difficult to use a server app than it would be to visit a web page, right? And another advantage is it can give you access to some really impressive remote computer resources. So that remote supercomputer where you're sending your data could be much more powerful than the computer you have on your desktop, right? The disadvantage, though, is that it does not provide that intellectual property protection. You're sending your data off to this remote server. And also, it can take a lot of effort to maintain this remote resource. So, maybe your funding runs out, or maybe someone decides they want to get a different job in industry where they can make 3 times more than they make in academia. This is something that happens, right? And suddenly you can no longer maintain this important resource to the community. And that has a big impact on reproducibility. And so my lab has really been pushing this, what we call a browser app approach to distributing tools for computer-aided drug discovery. And so in the browser app approach, you still you go to your web page, right? Your browser. But what happens is that the remote server sends analysis software to your browser. And then it's the same situation there in your browser. You load your data into the browser's memory, and you specify the parameters to control the calculation there in the browser. But then the calculation happens there in your own web browser. So it's not happening on a remote server because the remote server is sending the software to your browser rather than you sending your data to the server, right? And then once the calculation's done running there in your local browser, then you can view and save the results. And so the advantage here is that because the calculations run in your own browser, you never have to send your data to that third-party server. So you have the intellectual property protection, but it's still just like visiting a web page, right? So it's user-friendly. And it's very easy to maintain one of these browser apps because it turns out that it's no more difficult to send analysis software to a user than it is to send text or images. And so just a standard web server is plenty powerful enough to host one of these browser apps because the calculations are happening not on the remote resource, but on the user's local computer, right? And so my team has created a number of these browser apps, and a lot of times we'll see that there's a need in the community, and so we'll create an entirely new tool. Here are some examples of browser apps that are new tools that my lab developed, right? But sometimes we'll see that there's already good software to do the task that we want to perform, and the problem is just that it's not designed to run in a web browser. It's designed to run from a command line. And so sometimes we'll create browser apps where we're taking tools developed by other people and then we adapt them for the browser environment. So we don't necessarily need to reinvent the wheel, right? Now, the problem with our current approach, having this constellation of many different browser apps, right, is that can lead to a lot of challenges. It's very difficult to use all of these in a single workflow. So let me just give you an example. Say you want to predict how a small molecule binds to a protein. Well, first you need to know where are the pockets on the surface of the protein. And you could use our FPocket web browser app to identify those pockets. But then you would need to download or save the location of the pocket, right? And then you take that and you could go, say you wanted to next predict how the small molecule might bind in that pocket. You could come to our Webina browser app, but you'd have to re-upload again your protein structure, and you'd have to re-upload your ligands, and you'd have to re-enter in the location of the pocket. And so it becomes very difficult to basically have a workflow where you can integrate all of these tools just into one single protocol, right? It's also very difficult to maintain all these different tools. Each of them have their own code base, which is very challenging. And then also, they each have their own user interface, and so it's inconsistent, and that also impacts usability. So really, we want to move away from this model of having lots of different browser app tools. And what we've done to address that issue is we've created this MolModa environment, which provides a single browser-based environment for all of our tools. And so it's still the browser app philosophy, right? All the calculations are still running in your local browser, but the different tools aren't separate browser apps. They are plugins within this one single environment. And so now we have that workflow and interoperability feature that we're looking for. You can run one plugin right here in MolModa. The results are put here in the central browser, and then you can just load those results right into the next plugin without ever having to leave MolModa, right? And it's a single code base with this unified user interface, and we think that this is really a much better approach for the end user than having a whole bunch of tools at separate locations. So initially, MolModa is a tool for molecular docking. We published a manuscript describing this in Nucleic Acids Research, but our future plans are to transform MolModa into something that's more than just docking. We also want to integrate in new features focused on visualization, molecular visualization, and lead optimization. All right, so I'm going to give a little advertisement here. You can use MolModa, the current version, at durantlab.pitt.edu/MolModa. I think this is the version that CT is going to present later on. There's also this beta version, which is where we're testing out our latest features. So I wouldn't necessarily recommend using the beta version for your research because it's still going to change, right? But if you just want to see what we're up to, feel free to visit this beta site as well. So here I'm just going to give you a brief overview of MolModa's key new features and focus on that beta functionality so that Dr. Juso can then give you the hands-on demonstration with the production version in just a bit, right? Okay, so I've introduced you to my lab. I've explained the motivation behind wanting to create these web-centric tools. Now I want to introduce you a little bit to what exactly MolModa can do in terms of molecular docking. All right, so here I think most people here are familiar with molecular docking, but just to make sure we're on the same page, molecular docking involves these two steps, where first you have a model of a small molecule, and you have the pocket on your protein, and the first step is to predict the geometry of binding. So how does this molecule sit within that binding pocket? What is its pose, its three-dimensional pose, right? And then the second step takes that pose and it maps it to some score that hopefully correlates with binding affinity. This allows you to assess how well this molecule might bind to this drug target. Now, it takes a lot of preparation, right? Before you ever get to the docking step, you have to prepare your model of a small molecule and prepare your model of a protein. And MolModa has some plugins that allow for that preparation step right there in your browser. And so first thing to do is to add hydrogen atoms to your small molecules. And this is really critical because your molecule needs to reflect biological reality if you're going to get a good prediction. And adding hydrogen atoms to carbon atoms is usually very straightforward, right? There aren't usually any radicals or anything like that in a small molecule drug. And so it's pretty obvious where you're going to put the hydrogen atoms when they're bound to a carbon. But when you're considering the oxygen and nitrogen atoms, it's a bit more complicated to determine whether or not to add a hydrogen because the pH there depends on the solution and on the local protein environment. So if your small molecule has a carboxylate group on it, well, at physiological pH, probably it's going to be deprotonated, so negatively charged. If it's a terminal amine, probably going to be fully protonated, so it's going to have a positive charge at physiological pH. And then there's, a lot of other chemical groups where sometimes the protonation is fairly obvious, but sometimes there could be multiple protonation states. And so it's a complicated question to determine where to add the hydrogen atoms to molecules like oxygen and nitrogen. Now, fortunately, there is free software to do this, and it's called OpenBabel. Now, OpenBabel is a popular open source library for converting between different molecules and for processing molecular structures. The problem, though, is that it's written mostly in C and C++. So this was never really meant, OpenBabel, for some reason its logo is a fish, I don't know why, but OpenBabel was never meant to run within a web browser. It was meant to run as a command line tool, right? And that's because in a web browser, traditionally, it's only been possible, the only programming language you can use in a web browser is JavaScript, right? And this is written in C and C++. But what others have done and what we've done also is compile OpenBabel to something called WebAssembly. So WebAssembly is this new technology that runs in the browser. And just as you can compile software written in C or C++ to run on Windows or Mac or Linux, you can compile it to run on WebAssembly, which is like a miniature operating system that runs in your browser, right? And that way you can get this code written in languages other than JavaScript to run entirely in the browser environment. Now, just because you can compile something to run in the browser doesn't necessarily mean it's easy to use, right? But then you can write, HTML and CSS and JavaScript interface that can run and retrieve information from that executable as if it were running it from your command line, right? So you can build graphical user interfaces and websites around that compiled binary. And so that's exactly what we did with MolModa. We created MolModa around OpenBabel, this OpenBabel executable. And so I thought just to give a demonstration here of why protonation matters, I've prepared here a MolModa session for us. So here we have influenza neuraminidase, and you can see that it crystallized in this case as 2 tetramers, right? Now what I'm going to do is I'm just going to go ahead and keep only chain A. So here in MolModa, I'm just going to delete all these other chains just to kind of simplify what we're looking at here. All right, and what I want to show you is we'll zoom in here on this compound. This is actually oseltamivir or Tamiflu. It's a clinically approved drug that binds to this influenza neuraminidase protein. And so if you ever get a very, very bad flu, you can take Tamiflu and it helps some with the infection, right? And you can see here on this oseltamivir, it has a carboxylate group. And the question is, should this be protonated so that it's neutrally charged, or should it be deprotonated? Or should we add a hydrogen atom, or should we not add a hydrogen atom? And if we come over here and look at the amino acids on the protein, first of all, you'll notice there's no hydrogen atoms here because this is a crystal structure and it's not high enough resolution to resolve the hydrogen atoms. But you can see right here, we have an arginine, which is positively charged, right? And if this carboxylate group were negatively charged, well, then you would have a salt bridge, right? But if it's neutral, because you've added a hydrogen atom, there would not be that salt bridge. So really, protonation here is critical for maintaining this electrostatic interaction that is so important for binding. And in fact, if you look over here, there's even a primary amine, and we run into the same situation. If we add 2 hydrogens, it's neutrally charged. If we add 3, then it's positively charged. But here we have right here this negatively charged amino acid. And so if we correctly protonate this so that it's positively charged, we'll have a salt bridge. If we don't, then we'll have no salt bridge. So anyway, this really illustrates how critically important it is to add these hydrogen atoms correctly for your pH. And so what I'm going to do is I'm going to come up here to compounds protonation, and I'm going to go ahead and press protonate at physiological pH. And so what's happening right now is it's just downloaded that WebAssembly compiled version of OpenBabel, and it added hydrogen atoms to this small molecule according to that pH. And you can see, in fact, it did not add a hydrogen atom to the carboxylate group, and it did add 3 hydrogen atoms to this primary amine. So we have a negatively charged group and a positively charged group as required for this to bind well to the neuraminidase receptor. Okay, so another thing that MolModa can do that will help prepare your molecules for docking is to convert them to 3 dimensions. So what we saw just now in MolModa, the Tamiflu molecule, the oseltamivir molecule, already had 3 dimensions assigned, 3-dimensional coordinates assigned to its atoms, right? And that's because it was a crystal structure. But a lot of times when you're doing molecular docking, you're actually not working with a 3-dimensional molecule, but with what's called a smile string. And a lot of times, for example, databases of millions of compounds might not want to store the 3-dimensional coordinates of every atom. That takes up too much space. And so they just represent it. They encode the molecule in a smile string, which tells you what all of the individual atoms are in the molecule and tells you how they're connected, but doesn't tell you anything about their position in 3-dimensional space. Right? And so what I'm going to do is just to illustrate this. So this here is a complete smile string. I've just copied it to my clipboard, and I'm come over here and go edit, paste, and we're just going to paste that smile string here into MolModa. Now I'm going to hide neuraminidase, and I'm going to hide the protonated oseltamivir, and then we're going to focus here on the molecule I just pasted. And you can see that even though I pasted a smile string, when we've assigned the 3-dimensional coordinates to the molecule, it looks a lot like a person, right? This molecule is actually called NanoKid. I really like molecules that are designed to look like other things. I think it's funny. But if we zoom in here on NanoKid's hand, you can see that with all these atoms really do have full 3-dimensional coordinates. It's not flat. And so converting your smile strings into 3-dimensional molecules is really critical for performing accurate docking. All right, so next step. So I've described how MolModa can help you prepare your small molecules for docking. The next step also is to prepare your protein for docking, because recall that protein didn't have any hydrogen atoms added to it. As a crystal structure, it lacked the resolution, high enough resolution to position hydrogen atoms, right? So the key is to add the hydrogen atoms according to the pH of the environment where the protein resides. So for the vast majority of proteins, they are in a location in the body, for example, that is physiological pH, the blood maybe, is about 7.4, but maybe your protein is located in a lysozyme, and there the pH is closer to 4.5, or maybe it's one of those few drug targets that are in the stomach where the pH could be 1.5, so that the pH will impact which hydrogen atoms you should add to your protein. But the fortunate thing is that for the vast majority of pHs that you would ever encounter in the body, the protonation states are pretty unambiguous. So for the amino acids that end in carboxylates, so this is aspartic acid and glutamic acid, they are almost always going to be deprotonated, negatively charged, right? And arginine and lysine, they're almost always going to be fully protonated, and so they're going to be positively charged. The tricky one, though, is histidine, because the pKa of histidine is about 6, and that's close enough to physiological pH of 7.4 that it's possible to get both singly protonated and doubly protonated histidine. So it's possible for histidine to be neutral or potentially to be positively charged. And there's another complication too when it comes to adding hydrogen atoms to proteins. So in something like a crystal structure, what you get is just this density, this electron density, right? And then you have to position the atoms within that electron density. And for most of this, the amino acid side chains, it's pretty unambiguous how to position them in the density because the shape of the density is unique and it matches the shape of the amino acid, right? But there's a few cases where multiple different shapes match equally well. So for example, if you have an amino acid that has a terminal amide group, so this would be, for example, asparagine or glutamine, you're trying to position that terminal amide into this density, well, are you going to put the hydrogen bond acceptor, the hydrogen bond donor up here in this lobe? Or are you going to put the hydrogen bond donor, a nitrogen, in the lower lobe, right? And is the acceptor going to go in the lower lobe or the upper lobe? It's not obvious just based on the density how to flip that side chain. But it really does very much matter because you could have some other amino acid or even small molecule ligand that has a hydrogen bond donor that points towards this lobe. And if that's true, you're really going to want the hydrogen bond acceptor to be in that upper lobe, right? And so you need to intelligently flip these side chains so as to optimize these hydrogen bond networks. And MolModa fortunately also has tools for intelligently adding and flipping, adding hydrogen atoms and flipping side chains. For this, it uses a program called Reduce, which was made by others. And here it's the same story where this Reduce was never meant to run within a web browser. But my team took it and we compiled it to WebAssembly, and then we built this whole graphical user interface around it within MolModa so that it becomes much easier to use. So let's go ahead here, and I'm going to, I guess first I'm going to bring back these other ligands. I'm going to make NanoKid go away. Actually, I'll just delete that. So now recall we have over here this protonated oseltamivir sitting there in the context of its binding pocket. So I'm going to come up here to Proteins, Protonation, and click on the Protonate button. And again, what's happening right now is MolModa just downloaded a copy, a WebAssembly compiled copy of FPocket to your browser, and then it's adding hydrogen atoms here in the browser. So all that calculation is happening in the browser, not on a remote server. And let's go ahead and hide the originals. And you can see now that all of these hydrogen atoms have been added to your protein amino acids. So it's really critical to correctly protonate and prepare the protein as well before docking. And then just really to stress this, you can see here this carboxylate group, for example, is deprotonated as you would expect for physiological pH. So it's negatively charged and is well posed or well poised to interact with this positively charged primary amine. Right? All right, so another consideration is you need to identify the binding pocket on the surface of the protein where candidate small molecule ligands might bind, right? And a good binding pocket has a lot of very specific characteristics. It tends to be, when you're talking about small molecule ligands, a binding pocket tends to be kind of concave and enclosed. But beyond just having that enclosed shape, it usually is lined, the pocket is lined by amino acids that have side chains that can participate in hydrogen bonds with the small molecule ligand or electrostatic interactions with the small molecule ligand. It's not just a greasy pocket with hydrophobic side chains, right? There are molecular features there that can enable binding with a small molecule ligand. And so a lot of times it's easy to find a binding pocket. Now, a good example of this is that very neuraminidase example, right, that I just showed you. If we come over here, let's look at this protein in surface representation. You can see that small molecule ligand is sitting right there inside a very well-defined pocket. And so if you have a crystallographic ligand sitting in a pocket, then you know that is a binding pocket, right? But a lot of times you might not know exactly where the binding pocket is. For example, it could be that you have a crystal structure, but it wasn't co-crystallized with any ligand. And so there might be multiple pockets and you're not sure which one is the primary pocket to target. It could be maybe it's a model developed with AlphaFold 3 or something and doesn't have a ligand there. Or maybe you do know where the primary binding pocket is, but you want to identify and study other pockets, maybe allosteric pockets, where ligands could bind and affect the activity at the primary pocket. So there's all sorts of circumstances where you might not know exactly where a binding pocket is located. And so MolModa also provides this tool for identifying the location of pockets on the surfaces of proteins. And here again, we didn't want to reinvent the wheel, right? So we identified this program called FPocket, which again was created by others. That's a binary program that's meant to run from a command line. And what we did is as that same story where we compiled it to WebAssembly so that we could use it there in the MolModa environment. And you say that, like we compiled it to WebAssembly, and it sounds like it's a very easy thing to do, right? But in fact, it's very technical and very challenging thing to do. But this WebAssembly technology does make it possible, right? So let's go ahead and return here to our beta version of MolModa. And what I'm going to do is I'm going to come up here to proteins, and then I'm going to click on detect pockets. And then we're just going to detect all the pockets we can find on the surface of this protein. And again, what's just happening is a binary copy of FPocket was just downloaded here to my browser without me having to install or do anything. And then it ran that calculation right here in the browser. And you can see now this protein is covered in many boxes that indicate the locations of any pockets that the algorithm identified, right? And so what I'm going to do is these pockets are actually they are numbered according to sort of prioritized pockets, right? Let's go ahead and just hide all of the pockets, but then we're only going to look here at the first pocket, pocket 1. And you can see that in fact that pocket is the pocket that covers the crystallographic ligand. So it got the correct pocket here. I'm going to go ahead and zoom in now on the ligand. Now in this case, we know exactly where the ligand is. So if we didn't have a co-crystallized ligand, we might just leave this pocket as it is, right? But since we know that this crystallographic ligand is sitting in the pocket, what I'm going to do is I'm just going to recenter this pocket right on top of the ligand, right? Because we might as well take advantage of the information we have there. And then I'm also going to make this pocket just a little bit bigger so that it better encompasses that whole binding pocket. I'm going to come over here also, and I'm just going to rename this, just so I can remember what it is, docking box, right? So here we've managed to identify the location of that pocket. So we talked about how it's very important that the pocket not just be a greasy pocket, but that it be lined by amino acids that can participate in specific interactions. I'm going to come up here to binding and go to interaction diagram. And what this does is it's going to analyze, actually uses a third-party server. So in this case, it is sending your data to a third-party server, right? But it's analyzed that protein ligand interactions. And here you see the oseltamivir here in the middle. And there's all these amino acids lining that pocket. They're able to form these very specific interactions with oseltamivir, including a lot of the salt bridges, right, that we already discussed. So this just highlights that it really is a good binding pocket. It's lined by these amino acids that really can participate in very specific interactions with candidate ligands. All right, so now that we've prepared, we're getting prepared the protein and the ligand, we're getting much closer to being ready to do our molecular docking. And again, just as a reminder, that's that step where you predict the geometry of binding, and then you map that geometry to some score, right, that correlates with binding affinity. But in practice, a lot of times, you might want to dock just a single molecule into your binding pocket. Maybe you've already identified a good ligand, and you just want to get a sense of its molecular mechanism. So maybe you just want to dock a single molecule, right? But a lot of times, what you do is you want to dock many, many, many molecules into a binding pocket to try to identify which ones are most likely to be true ligands. And so when you have a molecular library like that, a lot of times you'll have some known ligands. So these are molecules that they bind in that pocket. And you might have a few of those, positive controls, right? But then you're going to have far more uncharacterized compounds. So it's not impossible that some of these molecules might bind in the pocket, but the vast majority of them almost certainly don't, right? And so you dock each of the molecules into that pocket, and then you predict their binding affinities, and you order the molecules by the binding affinity from the very best to the very worst, right? And in this case, since this is a made-up example, of course, the known ligands, right, are right at the top of that ordered list. And that's important because that tells you, those are positive controls, that tells you that your docking protocol can effectively identify true ligands. And then you can ask yourself, well, what about this ligand? This was one of the uncharacterized compounds. We don't know for sure that it binds to this protein, right? But it has a docking score that's similar to known binders, and our overall protocol did a good job of putting the known binders at the top of the list. So chances are this molecule, it's much more likely to be a true binder than just a molecule we would pick at random, right? And so in practice, you might pick like the top 20 or 40 molecules, whatever your experimental collaborator can handle, and you ask them to test them experimentally, and maybe of those 15% will be true ligands. And that doesn't sound like a great hit rate, but if you were to just pick molecules at random, your hit rate would probably be something like 0.1 to 1%. This is just a way of recommending molecules that are far more likely to be true binders than just molecules picked by chance, right? Okay, so let's return then to our neuraminidase example. And what I'm going to do is I want to create now a little compound library for us. So the first thing I'm going to do is I'm going to go to this PubChem plugin, and I'm just going to type in here peramivir. Peramivir is another molecule similar to oseltamivir that also binds to influenza neuraminidase. So this will be a second positive control in addition to oseltamivir. And then what I'm going to do is collapse this. I'm just going to hide peramivir for now, right? And I'm going to come up here, go to, compounds focus library. And here I'm going to pick, again, search PubChem for molecules that are very similar to oseltamivir. And I'm just going to pick 10 molecules that are similar for the purpose of this demo. And what this does is it searches that PubChem database, which is a public database, a repository that has a lot of information about many, many different molecules, right? And so it's search PubChem to try to identify molecules that are similar to oseltamivir. Let's go ahead now. See, all these molecules have been put over here kind of floating in space, and that's because in this case, oseltamivir, it was the co-crystallized ligand, so it's already positioned in the binding pocket, right? But for these new molecules, they're not docked or co-crystallized, and so it's just kind of automatically positioned off in space. Now, one thing to notice here, here we have in this ball of molecules a carboxylate group, right, that is fully protonated. So this is a neutrally charged carboxylate group. That is not how carboxylates look at physiological pH. So the protonation here on these small molecules is not correct. But we now do at least have that library of molecules that we can consider. What I'm going to do then is I'm going to come up here to compounds. Let's reprotonate all of these. Now you'll notice there's 12 compounds here. One of those is the oseltamivir, right? And then the other one is the peramivir we added. And then there's the 10 molecules that are similar to oseltamivir. So let's just go ahead and protonate all of these. And I'm going to hide all of the old versions of these molecules so that we can just look at the new protonated ones. And right off the bat, you can see here's a carboxylate group, and now it's negatively charged. So it's correctly assigned hydrogen atoms to these 12 molecules in our library. All right, so now we've prepared our protein, we've prepared our small molecules, we have identified a box where we're going to dock our molecules. We're now ready to begin the docking step, finally, right? And so MolModa uses a WebAssembly compiled version of Autodock Vina to do docking. Vina is very popular docking software. It's free and open source, written in C++, but it's that same issue of it's a command line program, right? And so it was never meant to run within a web browser, but my former lab programmer, Yuri Kochnev, did manage to compile it to WebAssembly, and we've now integrated it into MolModa. All right, so let's go ahead and show this doc. Now I'm going through all these docking steps very quickly, right? But that's because Dr. Juso is going to give a more thorough a demo after, right? So I'm going to go here and do compound docking scoring. Right off the bat, I'm seeing 0 proteins, right? And that's because I forgot. We need to make sure our protein is visible. And actually, you know what? That is the unproteinated protein, so that's not what we want. Let's hide all the unproteinated. And actually, we don't need to include the crystallographic oseltamivir because it's already included down here. All right, so now we have the proteinated protein and we have the proteinated molecular library. Let's go back here to perform docking scoring. Now you see there's 1 protein, 12 compounds. That's what I was hoping for. We're going to select that docking box that we identified previously, and we'll go ahead and press this dock button. And so what's happening right now, again, is that WebAssembly compiled version of Autodoc Vina has just been downloaded to the browser, and it's now performing the docking calculation right here in the browser. If we come down here, we can see, I'm going to expand this just a little bit so you can see better. You can see it's already docked 5 of these molecules. Let's go ahead and zoom in on them. And you can see that they are being positioned here within that docking box and within that binding pocket. In fact, I'm going to go ahead and get rid of this surface. And I'm going to also get rid of the individual atoms to make it easier to look at. And I'm also going to hide, whoops. I'm also going to hide this docking box. All right, so you can see then that all those molecules, those 12 molecules, have been positioned here within the binding pocket successfully. All right, so now that we have docked them, we come over here to the data table, right? And each entry over here has a whole bunch of data associated with it, but what we can do is we can hide all the data that isn't a docking score, right? And then we can go over here and we can sort these from the very best, the compound with the very best docking score to the very worst. And you can see that the compound with the very best docking score is peramivir, which is one of those positive controls we included in our screen, right? And oseltamivir is also at least midway through the list, right? This indicates that overall our docking protocol is pretty good at identifying true ligands. And so then the question is, well, what about these compounds right here, right? These compounds aren't known to be true ligands, but they have docking scores comparable to a known ligand in an overall screen that did a good job of prioritizing the known molecules. So these would be good candidates for future testing. All right, so I gave you a very quick demo of docking in MolModa. Again, I went through it very quickly, so CT will do a much more thorough job in her demo, going through it slower so that you can all understand it, right? Now I just want to conclude my talk by briefly discussing some future directions. Our future efforts are going to try to integrate lead optimization tools into DeepFrag. And so I'm going to go through this a little bit quicker than I was planning. I want to make sure that we know we're on the same page when I talk about lead optimization. So when I say lead optimization, I mean this process of iteratively adding or swapping fragments to improve a small molecule, especially to improve the binding affinity of a small molecule, although you can optimize for other properties as well. So here's an example targeting cyclin-dependent kinase that I found in the literature. These folks initially found this fragment that bound to CDK with a very high IC50 value, so not really very drug-like, right? But they discovered that if they added this fragment to that core scaffold, the IC50 would improve to 3 micromolar. And then if they swapped out this bicyclic moiety with just one ring, well, it would improve further to 97 nanomolar. And they just iteratively modify this molecule until eventually they've transformed this very weak ligand into a molecule with an IC50 of 47 nanomolar. And that's the kind of IC50 you would expect of a typical clinically approved drug, right? So it's this iterative process of improving binding. And so my lab has created a tool, a machine learning tool, to guide this lead optimization process. Here I'm showing you, it's called DeepFrag. Here I'm showing you the cover art for Chemical Science, where we published DeepFrag, to really brag a little bit about this fellow Harrison Green. So Harrison Green was an undergraduate in my lab at the time, and he really took the lead on the DeepFrag project. And it's just remarkable that an undergraduate could have played so prominent role in a project like this. And he also created this cover art. So he's a brilliant scientist as an undergraduate and also a great artist. I could not have put together something like this, right? So if you ever have a chance to meet Harrison Green, I recommend talking to him, right? So how does DeepFrag work? Well, DeepFrag predicts fragments that should be added to positioned or posed molecules. So you start with the structure of your protein, and then you start with the structure also of your small molecule, and it needs to be bound to the protein. So it's not a docking program, right? It needs to either be docked or co-crystallized. And then you indicate the location of an atom on that ligand to which you wish to add some optimizing fragment. And then DeepFrag will predict a topological fingerprint, just a series of numbers that describe the fragment that it thinks you ought to add at that location. But the tricky thing is this topological fingerprint doesn't tell you what the structure of the fragment is. It just describes the structure. And so we have this separate database, which we call a label set, of many, many small fragments. And we've gone and we've pre-calculated all of their fingerprints, right? And so we can take this DeepFrag predicted fingerprint and we can compare it to each of these pre-calculated fingerprints. And then we can identify the one that's most similar to the DeepFrag prediction. And then we have the structure, right? Because we've pre-calculated all this. And we can take the structure of that fragment and we can add it back at the originally specified user location. And the hope then is that this composite ligand, the parent portion of the ligand plus the fragment, will be a better binder than the original parent ligand was, right? And so how do we capture the structures of the protein and that original ligand? Well, what we do is we voxelize them. So basically for each of the atom types, we create a separate grid of 24 by 24 by 24 points that are each spaced 0.75 angstroms apart, right? So for example, on all the carbons of the protein receptor, we would map all of them to one grid, and then maybe all of the oxygens on the receptor would be mapped to another grid, and all of the nitrogens on the ligand would be mapped to another grid. So we have a series of many grids that each capture the positions of the atoms, different kinds of atoms on the protein and the receptor. And so how do we create these grids? Well, here's a very simple example, right? Say we want to map these atoms onto these discrete grid points. Well, you can see here, this particular carbon is very close to one of the grid points, right? And so we would put the value at that grid point very high because of how close it is to a carbon. Now consider this grid point. It's sort of close to carbons, right? But not very, very close. And so we would assign some value there, but it wouldn't be as high as the ones that are close to carbons. And then, for a grid point that's far away from any atom, well, we wouldn't really assign any value there. So this discrete grid captures the locations of each of the carbon atoms, right? And you could then create a grid for the oxygen atoms and a grid for the nitrogen atoms. And so just imagine the same thing, except instead of a flat molecule, it's a three-dimensional protein and ligand, and instead of a two-dimensional grid, it's a three-dimensional grid. And that's how we voxelize those different atom types, right? And so we take all those voxel grids and we concatenate them into one big tensor, and we feed that into this DeepFrag model. And when DeepFrag hasn't been trained, it'll initially just kind of output random numbers that don't really correspond to anything meaningful. But what we do is we take these molecules, like thousands and thousands of molecules that are known to bind to this protein, and we cut them along a single bond, right? And then we provide DeepFrag with the parent portion of the molecule as a voxel, but we calculate the fingerprint of the fragment, right? And then what we do is we compare the DeepFrag prediction to the actual correct fingerprint, and we can calculate the difference between them. And we just keep then training this, we keep training DeepFrag until its predictions closely match the actual true fragments, right? So we don't do this with just one protein ligand complex, but we do it with tens of thousands, hundreds of thousands of complexes. And so DeepFrag sees many, many examples of true fragments And that's how it's able to learn these sort of general principles of fragment addition. All right, so to train DeepFrag, it's pretty intensive. We have to do it on a very high-end graphical processing unit for about 5 days, right? But once it's trained, the good news, and say you just want to apply DeepFrag to a single protein ligand complex. Once it's trained, it's actually not very computationally intensive to use at all. You can make a prediction on a single protein ligand complex in about 30 seconds on a standard CPU. You don't even really need a GPU, right? So we tested how often DeepFrag would predict the correct fragments. So what we did is when we created a label set that always included among the many, many options the correct fragment. And then because we use that label set, we were able to ask the question, how often is the label set fragment closest to DeepFrag's prediction actually the correct fragment? And so that's what this top one accuracy is. And there we got a top one accuracy of about 58%, right? So about 58% of the time, DeepFrag got it right. Now the correct answer was among the 8 most similar label set compounds, 67% of the time, and among the top 64, about 74% of the time. So it really suggests that DeepFrag has learned some of these core principles of lead optimization. We're also curious if DeepFrag was using information about both the protein and the ligand to make its predictions, because remember, both of those are voxelized. And so we created a separate DeepFrag model where we only provided the voxels of the ligand, the parent portion of the ligand, right? And it still got the right answer about 48% of the time. Then we did another model where we only provided it with the protein, and it still got the answer right about 38% of the time. So it's impressive it did so well when it only had some of the information. But the important thing to note is when we provided it with both the ligand and the protein receptor, it was far more accurate than when we provided only one or the other. So that really suggests that DeepFrag has learned to leverage information about the entire context, both the protein and the receptor, making its prediction, which was exactly what we were hoping for, right? Remember, I mentioned that DeepFrag is, to train it was very intensive, but to use it is really not, it doesn't require a lot of computer power at all, so much so that it can even run in a web browser. And so it's the same story where you load your receptor into your browser's memory. You never send it to a third-party server, just load it into your browser's memory. You load the structure of your ligand into your browser's memory, and then you select which atom you want to, select the atom to which you want to add some fragment. And then you press that Start DeepFrag button, and it then downloads the DeepFrag model to your browser and runs it entirely in the browser, and then makes its fragment recommendations without you ever having to send your data to a third-party server. And so we're very pleased to learn that DeepFrag is efficient enough that it can run in a browser. The problem is we've not yet integrated this into that MolModa environment. And so that's really the next steps. We want to get lead optimization tools like this, as well as other lead optimization tools as plugins within MolModa, so that you can have your initial ligand, you can add fragments to it intelligently, and then you could dock those optimized molecules all right there in the same environment using a single unified workflow. All right, so I'd certainly like to acknowledge all the members of my lab, both those who have graduated or gone on to other positions, as well as my current students. I'm very grateful for all the help they've offered on projects such as these. And I'd certainly like to acknowledge these important sources of funding. So very happy to answer any questions, and thank you very much for your time.